Hello student, very good morning to all of you. How are you? I hope you all are happy, healthy and fine at home. Do you remember what were we discussing yesterday? I think we were discussing about the glycosidic phosphate method of glycosylation or the glycosylation method use of glycosyl phosphate. How to prepare glycosyl phosphate and what are the uses of it, or how we can synthesize or how we can derive the derivatives of that phosphate, as well as how we can use it, or what are the application of glycosyl phosphate with the help of. There are some method for the synthesis of glycosyl phosphate, and by using that glycosyl phosphate how we can derive different type of the derivatives that we have discussed in detail with the suitable example. So now today what we are going to discuss, let's look at here, because we are discussing the point synthesis of glycosides, that is a B point. Here glycosylation methods means use of N-pentenyl glycosides. First of all, let me tell you how to remember these derivatives, or how these derivatives can be used for the synthesis of another derivative, means application of this derivative, how it is utilized in the higher derivatives, which is biological application of that derivatives means. And let me tell you, whatever this derivative N-pentyl glycoside derivative, from where, what are the starting material, how it is prepared and how easily it is prepared, why it produced as a high yielding product. Once you get this high yielding product, that N-pentyl glycoside derivative, how we can convert, how we can obtain another derivative. In that way you have to remember, you have to discuss, you have to study this type of the compound over here. Now let us see here, glycosylation method means use of N-pentyl glycoside. Let us discuss some introduction regarding this N-pentyl glycosides. What do you mean by the N-pentyl glycoside, or how do we define N-pentyl glycosides? What are the method for the synthesis of N-pentyl glycosides, and how these different derivatives can be converted into another derivative with high yielding, and all are the stereoselective reaction at, or what we can call it as a anomeric controlled reaction, high yielding product is obtained. The use of protecting groups in synthesis of complex oligosaccharide is currently inevitable. Protecting group here, there are number of hydroxyl groups are present. If you observe the structure of carbohydrate, according to its definition, carbohydrates are polyhydroxy aldehyde or ketone. On hydrolysis gives this type of the compound are known as a carbohydrate, and therefore protection of hydroxyl group with specific or the suitable protecting group, the good yield of the protecting group or blocking of that functional group. There are number of functional group, mainly in a carbohydrate, primary alcohol and the secondary alcohols are present. By using the specific or the suitable protecting group, which will be, as we know that the protection or the deprotection, it is an additional step involved, but it is a necessary. If it is not protected, then we cannot get the desired product, and for to obtain the desired product, protection is required or protection is needed, and that protecting group should be installed, or that groups, functional groups are blocked in a mild condition with high yielding, and whenever we require, we can deprotect it, and during that reaction condition, as we know that oxidation, reduction, reaction are carried out in acidic medium, basic medium. So whenever we are protecting up the group, that may be sustained or remain stable in a particular medium or at a reaction condition. Nothing will happen when we are carrying out, when we are converting the other functional group by adding the suitable reagent. We can convert one functional group into as another desired product. Once you get, we can deprotect it in a mild condition. So that are the some of the criteria, as we already learned, for the protecting group. Electronic and steric factors may enable radioselectivity. As we know that radioselectivity, it is a stereochemical term involved here. Radio means at a particular reagent, at a particular region, the reagent is attacking, and we are getting that specific or desired product. So that type of the reagent or that reaction are known as radioselective reaction. For example, Wittig reagent. If you remember, do you remember the Wittig reagent? Wittig reagent is used to, or Wittig reagent to be used to convert the carbonyl group into olefin. Means, suppose if you take an example of cyclohexanol treated with Wittig reagent, the product obtained will be the exocyclic double bond over there. So at a specific position only, double bond we can getting with high yield, and that kind of the reaction are known as radioselective reaction. So glycosylation is the reaction in which a carbohydrate, that is a glycosyl donor, is attached to a hydroxyl or other functional group of another molecule, a glycosyl acceptor, hydroxyl groups, but for the foreseeable future, such interconversions will be exception to the rule. If protecting groups are to be necessary it would be advantageous for them to serve additional roles. In our laboratory, this concept was first appreciated with the development of N-pentyl glycoside, NPG. Remember here, this is in short is known as a NPG. Remember, not the NGP. NGP means was neighboring group participation, but here NPG means N-pentyl glycoside. So do remember this acronym here, NPG, in which the in our laboratory this concept was first appreciated with the development of N-pentyl glycoside, NPG, in which the pentyl group protect the sensitive anomeric. Sensitive anomeric, let me tell you, anomeric means that carbon number 1 or it is next to the oxygen when you move clockwise manner, then remember, or that anomeric carbon atom, it is additional chiral center is formed when there is a ring formation of carbohydrates are generally as the simplest one is a D-glucose. That pentenyl groups protect the sensitive anomeric center, which is very sensitive and survives most of the chemical transformations tolerated by normal alkyl glycosides. Treatment with an electrophile converts the pentenyl moiety into a living group, however, thereby enabling replacement reaction to occur at the anomeric center. The dual means two. The dual protection and activation role is a fundamental property of NPG, and this duality was heavily exploited in the synthesis of the rat brain, we know that, mouse, rat brain, Thi-1 glycosylphospholidylacetal membrane anchored by the retrosynthesis plan outlined in scheme number 1, that is a retrosynthetic analysis. As we know that retrosynthetic analysis means what? The process of breaking down target molecule into convenient starting material is known as a retrosynthetic analysis. And as we know that retrosynthesis, RS, retrosynthesis, an imaginary but logical reverse chemical reaction which help us to reach the reactant starting from the target molecule is known as a retrosynthesis. And where D connection, FGI, these are the some of the important terms used in retrosynthetic analysis. Disconnection means what? An analytical operation which break a bond and convert a molecule into a possible starting material is known as disconnection. And there are special arrows, if you remember the special arrows which we have learned during that chiron approach, where you have learned this type of the terminologies. Or and FGI means functional group interconversion. One of the functional group is converted into another functional group so that disconnection becomes possible is known as a functional group interconversion, and FGI written over the disconnection arrow. Again, there are certain terms are used like as a synthon, synthetic equivalent, reagent. Synthon means what? After disconnection, whatever positively or negatively charged species are obtained or fragmentation are known as a synthon. and these synthons are very unstable, and it must have a synthetic equivalent, means the function carrying out the function of synthon, the reagent carrying out the function of synthon is known as a synthetic equivalent. And the reagent means what? A compound which gives rise to an intermediate, or the target molecule itself is known as a reagent. So these are the terminology, we are not going to go into detail, but now here, so let us see here how we get this one big molecule, that is a thione 1, answer rat brain. So that I am trying to draw over here. Let us see, this is the compound. Let's see here, this is one of the compound, then oxygen atom, and then this group. Then we can draw over here this one, then next one will be oxygen atom, then this group. Again one can draw here then this type of the compound, oxygen, then this or this one we can show like this. There will be this type of the compound, once again oxygen over here, this one you can draw like this, and finally is a here in the last ring there is no oxygen atom, remember, OH, this is a structure number first, here may be as a, this is a structure number first, then this will be the structure number second, this will be the third, fourth, and the fifth one. So likewise, so second one, here in the second I am showing this one. Now in the first one we can show this is a equatorial oxygen atom, then this bond, then there will be P, then we can show double bond O, hydroxy group, then here is a oxygen atom, then let me show here once again there is a oxygen atom, then C double bond O, then we can show 8 times like this, 1 2 3 4 5 6 7 and 8 times. Similarly, once oxygen atom over there, again 1 2 3 4 5 6 7 8 9 and 10, this one, and in the second this one, third one is this one, where here is again oxygen atom, then here will be phosphorus, OH, and next one is NH2, and here we can third once again here equatorial position, oxygen, again here you can show this one, there will be a, here is the oxygen atom, then we can show like this. Here once again at this position, I think this is not above one, this is a, let me clear this point once again over here. Here is the oxygen atom, then this one, then you can show like this, then there will be a hydroxy group, then next one at axial position hydroxyl group, equatorial position hydroxyl group, and now come to the fourth one, hydroxyl group here, hydroxyl group at this position, and now at the fifth one, fourth one, this one, and now once here is a hydroxyl group, then fifth one, once again you can draw here is a oxygen atom, then phosphorus double bond O, hydroxy group, then you can draw here oxygen, further here like this, then there will be NH, then peptide type of the bond, and here is a protein, as we know that protein is made up of amino acids, and here is a this one hydroxy group, once again here is a hydroxyl group, here also as a hydroxyl group, and here once again as a hydroxyl group. So this is the big structure. So this structure, the name of this is a, this figure indicate as THY, Thyone, Thyone anchor, rat brain, BRAIN, rat brain. So that is a name of this complicated, very complicated molecule, and once it is shown as a this, as we know that this is a retrosynthetic arrow, and that becomes into as a three part. So that three parts, first, second and third part are shown over here. First one which is again as this scheme number 1, retrosynthetic plan for rat brain Thyone, GPI membrane anchor is shown over here. So how it will be, let's see structure number first and the second, how we can show over here. So let's see this one. Now there is a no oxygen atom involved. This is structure number first, OBN. Here is OBN, and at this equatorial position here O, then there will be this one allylic double bond here, and another OBN over here, OBN benzyl group protection over there, and now at this position there is an oxygen atom, and then it will be at axial position. Here axial position there will be this one oxygen atom over here. This we can show like this. So here this is a structure number second, this will be the first one. Then here we can show OBN. Here is a hydroxyl group which is free for desired glycosylic bond formation, OBN, this one OBN, This is the structure number first and the structure number first and second up to this one, and now is a third this one we can break up to this one, and then fourth and fifth like this retro we can show. So this is a retrosynthetic plan for rad brain Thi 1 GPI membrane answer, that is the scheme number 1 is shown over here. So this is structure number first. So second we can draw the structure like this, so we can get this type of the compound. So I think so fundamental reaction, a summary of most of the fundamental reaction of N-pentyl glycoside is presented in scheme number second, and that are the scheme number second shown as how it can act as a electron donor as well as electron acceptor with the general reaction. I think with this we have seen this is one of the how N-pentyl can be used. I think in the next time we will see the remaining point of this synthesis of glycoside. I think with this let us stop today here. Remaining we will see the next lecture. Thank you. Have a nice day.